Good morning, Professor. My name is Anudaya and my partner's name is Manveer Singh. Today, me and Manveer is going to represent a presentation and the name of the topic is Total Microbial Load and Pathogen Screening in different type of lettuce and to determine the effectiveness of different disinfectants and UV effect on lettuce. In the second slide, I am going to talk about the introduction. As we all know that people consume lettuce as a raw green vegetables as it is a healthy food and contains nutrient rich diet and a lot of minerals in it, and it also protects us from disease such as cancer, heart disease and stroke. Lettuce leaves are highly sensitive to bacteria and there are several types of bacteria present in lettuce leaves such as E. coli, salmonella, listeria, asorius and fungi. In the third slide, I am going to talk about the source of contamination in lettuce. The primary source of contamination is soil, as the vegetables are grown in soil. The secondary source of contamination is manure and water, as it is required for the nourishment of the vegetable. And the last is the processing and transportation, from which the bacterial contamination occurs. In the next slide, I am going to talk about why we use the lettuce, as it is consumed as raw or uncooked and it contains several types of bacteria which are toxic to health. E. coli O157 Shiga, a toxin produced by H7 which kills the red blood cells and causes renal failure and bloody diarrhea. Also, the other strain of E. coli causes several diseases such as pneumonia, lung illness, and urinary tract infection. And also, there are Asorius, Salmonella, Listeria, and fungi, which are also toxic to the human health. And the objectives of this experiment is to evaluate the total microbial load in different type of lettuce such as iceberg and romaine lettuce, to isolate the colonies and identify the concerned pathogens if present, to evaluate the non-invasive disinfection method and their efficacy in lettuce and with the application of UV treatment. The hypothesis of this experiment is that iceberg lettuce is more contaminated with E. coli bacteria and has a higher microbial load than romaine lettuce. And the sodium hypochlorite is more effective disinfectant for lettuce than hydrogen peroxide or UV treatment for microbial production. So now I'm going to talk about materials and methods. Materials are romaine lettuce, iceberg lettuce, pipettes, bulb, beaker, hot plates, and knife, phosphate buffer, TSA, MSA, EMB plates, and for the preparation of TSA we required media, and for EMB the EMB media is required, and for the MSA, the mineral salt agar is required, and also the stirrer. So the first step is the preparation of iceberg and romaine lettuce sample. So firstly, took out the inside and outer leaves of iceberg and romaine lettuce and cut into pieces separately and weigh 25 grams for each type of lettuce. And after that, put it in stomacher bag and pour 225 ml phosphate buffer so that we can make 1:10 ratio in both the lettuces and put in the stomacher bag. After that, mix the sample in stomacher bag for 2 to 3 minutes and leave it for 20 minutes. After that, took out the 5 ml sample in the dry test tube and the serial dilution is done up to 10 to the power minus 8 for both the sample. 0.1 ml from each test tube were poured on TSA plate and then spread it on the TSA plate with the spreader for both the lettuce sample test tubes. And the last step is to incubate the TSA plates at 37 degrees Celsius for 24 hours. The second step is the preparation of lettuce sample with disinfectant, and my partner is going to explain that step. Thank you.
Good morning, Professor. I'm the second partner in the experiment. So as Anu discussed about the sample preparation before the disinfectant, and I'm going to discuss the second part in which we prepare the sample with the disinfectant such as sodium hypochlorite and hydrogen peroxide, and also with the UV treatment. So we prepare the sample by taking the 25 gram of a sample and dipped into the different disinfectants such as sodium hypochlorite and hydrogen peroxide, and also treated with the UV on the same time. And after that we rinse both the samples, and then put it in the stomacher bag for each sample, and put it the 225 ml of the phosphate buffer. And after that we mix it in the stomacher bag for 2 to 3 minutes, and then we did the 10 serial dilution till 10 raised to power minus 8 for the both the samples which were treated with the sodium hypochlorite and the hydrogen peroxide. And for the UV, we take the 2 ml in the petri dish and put it under the UV radiation lamp for the 2 to 3 minutes. After that, we did the same serial dilution as we did with the two different type of disinfectant till 10 raised to power minus 8. And after that, we put the 0.1 ml poured into the TSA plate and incubate it for 37 degrees Celsius for 24 hours. And in this we can see the results which were obtained after the before disinfectant. So from the this table we can conclude that the iceberg lettuce has a more contamination. There was a more microbial growth as compared to the romaine lettuce. And the growth in the positive, there was a growth was observed in the positive controls, and then there was no growth observed in the negative control for the both the different type of lettuce. As we can conclude from the photos also, there was a iceberg lettuce has a more contamination as compared to the romaine lettuce. And we did the biochemical test and the gram staining also. As from the oxidase test we confirm we didn't get the positive results, so there was no purple color observed. So both the tests for the both the plates were negative. And the catalase test, there was a positive test as there was a positive results, as there was a formation of a bubble on the plate. And then we further did the gram staining, and from the gram staining for the both the plates we conclude that the one plate, for a plate 1 there was a gram negative bacteria which were rod shaped, and for plate 2 there were the gram positive and have a cocci shaped cells. So, and then after that we did the confirmatory test for the bacteria, as we take the EMB plate or the MSA plate. Or on the EMB plate we observed the uh, green metallic sheen after the incubation of 37 degrees Celsius for 24 hours. And that confirms, the green shield confirms the presence of E. coli. And on the MSA, there was a color change after the incubation of 37 degrees Celsius, and that confirmed that the presence of S. aureus. As a high salt concentration inhibit the growth of most gram-negative bacteria, and S. aureus mainly ferment the mannitol. So thus it confirms the presence of S. aureus in the slide on the MSA plate. And we also did the enteroplory test, from which we conclude that the code we observed in the code book and the bacteria found was the enterobacter. And the results for this rapid step plus reagent were not able, we were not able to analyze them because of the over incubation. And in this slide I'm going to discuss about the overall experiment. In this way, this table represents the data from the before and the disinfectant, after disinfectant with the two type of, and then with the UV treatment. And from this table we can conclude that the UV treatment is more effective as compared to the other treatments for the for killing the bacteria, or for disinfecting the lettuce sample. So we can conclude that the UV is the best among the disinfectant treatment. And for the positive, all in all among all the methods, the positive controls for the used for the before disinfectant and with sodium hypochlorite, UV, and hydrogen peroxide were uh, positive. And in the negative there was a no growth, and in the positive there was a growth. And these were the plates which were used as in the table 5 I discussed before and after, and the UV treatment. And the way for the identification we also did some test. I'm going to discuss with the 2-3 test from this, as the coliform test tube test which confirms the presence of E. coli. In the same way we did the TABC plate test, in which we conclude that the growth was seen as the purple color, which confirmed the presence of E. coli. In the same way we did the 3M petri film test. From there the pink color colonies with air bubble were seen, which also confirmed the presence of E. coli. And in the end, I'm going to conclude that the iceberg lettuce has a more microbial load than the romaine lettuce, and E. coli and Asorius are present in the lettuce, and UV has a more efficacy to kill the bacteria in the lettuce among the sodium hypochlorite and the hydrogen peroxide. Both gram-positive and gram-negative bacteria are present in the lettuce sample, which are toxic to the human health. And the rapid step plus a reagent test we also did, but due to the over incubation, we didn't get any results. And from uh, mm, the enteroplurid test, we conclude that it has an enterobacter species. So that's all from my side. Thank you.